Oh, hi everyone. Thanks for joining us today. My name is Julia Warrick. I'm in Mike Kearse's lab, and I'll be the moderator for this session today. So first, we just wanted to highlight all of the participating centers for the seminar series. And then also just this reminder and that the talks will be uploaded to the YouTube channel shortly after the session, which is again shown here, the YouTube channel. And then today, this RNA collaborative is being hosted by the Center for RNA Biology at Ohio State University. So we've been an official university center since 2012, and the center now has 45 member labs, which focus on all aspects of RNA biology. So today's talks will be presented by one postdoc and one faculty member. And so I'll go ahead and introduce our first speaker, which will be given by Dr. Gina Nostramo. Dr. Nostramo received her PhD from New York Medical College, working on the effects of stress on the renin-angiotensin system. And then she came to OSU as a postdoctoral fellow in the lab of Dr. Paul Herman, where she worked on ribonucleoprotein complexes. And she's currently a postdoctoral scholar at OSU in Dr. Anita Hopper's lab. So Dr. Nostramo has published in many top journals, including Nucleic Acids Research, Genetics, and she has a paper under revision in Molecular Cell. And she's also received many awards throughout her career, including a Pelotonia postdoctoral fellowship, several poster awards, and she's been an invited or selected speaker at many different conferences. So today, Dr. Nostramo will be sharing her work on free introns of tRNAs as complementarity-dependent regulators of gene expression.
Great. Thank you so much for that introduction. And thank you for the RNA Collaborative for the opportunity to speak today and share some of the work that we've been doing in the Hopper Lab. This work is currently unpublished, but is, as Julie mentioned, is under review. So hopefully it will be out in the next few weeks or so. Okay, so we all know that tRNAs function as adapter molecules in protein synthesis, and they carry amino acids to the translating ribosomes to decode messenger RNA into protein. And so in addition to this canonical role, we know that tRNAs serve many other functions, and as a result, defects in tRNA biology have detrimental impacts to human health and can lead to diseases such as neurological disorders and cancer. Now, in recent years, there's been a resurgence in tRNA research, which has brought into focus the emerging complexity of tRNA biology. And so if we think about the complexity of tRNA biology, it begins with the fact that there are many, even though there's only 20 amino acids, there are many tRNA encoding genes in every organism. And so for humans, for example, there are about 416 tRNA genes, and then another about 100 tRNA-like genes. If we add to that the fact that we know the genetic code is degenerate, right? So for example, there are 3 anticodon sequences for tRNA alanine, which make up about 34 of the 416 tRNA genes in humans. Next, we can think about complexity as far as tRNA modification. So tRNAs are the most modified RNA in the cell. There are over 100 known modifications that are found on tRNAs, and there are about 13 modifications on average per tRNA. And then what adds to this complexity is that tRNA modifications don't happen in an all or none manner, that there are these microspecies that have different combinations of modifications. Okay, the next layer of complexity involves the myriad of proteins that interact with tRNAs. So this includes 3 pathways for precursor tRNA nuclear export from the nucleus to the cytoplasm, 2 of which were recently discovered in the Hopper lab. And each of these 3 pathways have preferences for different families of tRNAs. We also know that there's a number of tRNA aminoacyl synthetases that are in splice variant forms that can interact with different sets of proteins. Perhaps the newest area of research in tRNA biology is the discovery of tRNA fragments. So we know that precursor and mature tRNAs can be fragmented in multiple different ways, leading to these fragments that play numerous roles in cell biology and disease. And what I want to talk about today is what we believe is a new level of complexity in tRNA biology, and this is FitRNAs, or free introns of tRNAs. So from archaea to humans, a subset of tRNA encoding genes contain an intron. This intron is located one nucleotide, 3' to the anticodon, and is shown here in yellow. And the intron must be spliced out in order to generate a tRNA that is functional in protein synthesis. Now, the excised intron is typically rapidly turned over, and it's barely detectable under standard growth conditions. And this splicing process is essential because all eukaryotes have at least one tRNA family in which all of the reiterated genes contain an intron. So the genome can't be fully decoded into the proteome without tRNA splicing. So since tRNAs are so abundant in cells, there's a substantial amount of resources that are dedicated to transcribing, splicing, and degrading tRNA introns. And so if we look in yeast, for example, there's about 600,000 introns, free introns generated per generation of yeast. And so therefore, the conservation of introns in tRNA genes suggests that they provide a functional advantage to cells. So what do we know as far as the functions of tRNA introns? So it's been shown that at the DNA level, tRNA introns can impact the ability of tRNA genes to serve as insulators for nearby genes. And it's also been shown that in archaea, they can protect against integration of viral genomes. If we look at the RNA level, it's been shown that tRNA introns can direct precursor tRNAs that have mutations in the anticodon stem loop for decay. And then they can also affect whether modification enzymes can recognize a precursor tRNA and prevent tRNA mismodifications. Now, I want to highlight that these functions are all mediated by introns that are within the context of the tRNA body and not the free tRNA intron. And then furthermore, these findings don't explain the recent findings that show that tRNA intron sequences are conserved. So it's been shown that for the tRNA tyrosine intron, so this was done by the Moderator group, they showed that a dozen different species of Drosophilids, which are separated evolutionarily by 50 million years, that the sequence of the tRNA tyrosine intron is conserved. So we wanted to know if the same thing was true in closely related yeast species. So a very talented undergrad in the lab, Paolo, looked to see if this was the case. And so what I'm showing here is the conservation of the tRNA ILU intron. And so this is the longest tRNA intron in Cerevisiae. It's 60 nucleotides long. And you can see by the highlighted positions that in the genus Saccharomyces, most of the sequences share greater than 90% sequence conservation to the Cerevisiae intron. And this is, despite the fact that these organisms have diverged from Cerevisiae several million years ago. Paulo also looked at other genuses of yeast, and you also see that there is sequence conservation, although less so than within the Saccharomyces genus, but these are species that diverged from Cerevisiae, about 100 million years ago. So based on these observations, there appears to be an evolutionary selection for tRNA intron sequences. And since selection infers function, we asked the question, do tRNA introns have a sequence-dependent biological function? And so to answer this question, we took two approaches. The first was a bioinformatics approach, and this was spearheaded by Paolo. Our second approaches were molecular and genetic approaches. And so we'll first take a look at the bioinformatics and then move on to the molecular and the genetics data. Okay, so what we did is we used the Saccharomyces genome database to look for open reading frames in Cerevisiae to identify regions that have complementarity to tRNA introns. And the criteria that we used were we looked for a minimum of 13 nucleotides of perfect complementarity, or 14 out of 15 nucleotides before allowing for any GU base pairing. And the mismatch could only be a substitution, not an insertion or a deletion. The reason that we chose this level of complementarity is semi-arbitrary. So it allowed us to identify genes that have complementarity that are at levels that are statistically improbable, but also yielded a reasonable number of genes that we could analyze both individually and as a group. So using this criteria, we identified 137 ORFs that have complementarity to the 26 different tRNA intron sequences. And so in this chart here, you can see the number of ORFs that we identified, and this is broken down by each of the intron-containing tRNA families in yeast. So yeast have 10 intron-containing tRNA families. And then below the names of each of the tRNA families, you can see the number that's in parentheses is the length of the intron. And so I just want to highlight that the number of complementary ORFs that we're seeing is not simply a reflection of the length of the intron. So we conducted gene ontology analysis for all of these ORFs that we identified, and we found that these genes were enriched in processes that include transcription by RNA polymerase II, DNA replication, organelle, mitochondrial, and chromatin organization, and meiotic and mitotic cell cycle. So our initial analysis looked for complementarity specifically within ORFs, but Paulo extended this analysis to include the 5' and 3' UTRs. And what he found was that, remarkably, the intron possesses more complementarity to ORFs than it does to 5' and 3' UTRs combined. And this is despite the fact that the typical UTR lengths in Cerevisiae are about 50 to 200 nucleotides long. So I want to draw your focus to the tRNA ILU intron, which I've highlighted here. So again, this is the longest tRNA intron in yeast, and it has complementarity to the most ORFs, with complementarity to 33 ORFs at the original criteria that we have set. And so for most of my talk, I'll be focusing on the tRNA ILU intron, but I just want to let you know that the bioinformatics data is similar for the tRNA trip intron. Okay, so on this slide, you will see the results for the ORFs that have complementarity to the tRNA ILU intron. So across the top, I have the sequence of the tRNA ILU intron. It's listed 5' to 3'. And then in the rows below, I'm going to show you the location of complementarity for each of the ORFs that we identified. So for example, you can see here that the gene GPM2 has complementarity. It has 14 nucleotides of perfect complementarity. That's at the 5' end of the tRNA ILU intron. And so when we look at all 33 ORFs that we identified, we see that complementarity spans the full length of the intron. And I want to just also highlight that 6 of these ORFs have at least 14 nucleotides of complementarity. So it's a step up from our minimum criteria. And again, this is before allowing for any GU base pairing. And so in the next few slides, I'm going to be referencing the 33 ORFs that met our original criteria, as well as these 6 ORFs that meet this kind of more stringent criteria. Okay, so once we had this information, the first thing that Paulo wanted to do was determine whether the amount of complementarity that we see was also found in the mature tRNA ILU. So in yeast, there are 2 copies of the tRNA ILU UAU gene, both of which have introns, and the introns in these 2 genes are identical in sequence, but the exons have small differences in sequence. And so we have this graph broken down by the 2 tRNA ILU genes. And so here what you can see is, despite the fact that the tRNA ILU intron is smaller in size, it has complementarity to more ORFs than the mature tRNA ILU, which we were surprised by. And this is even more apparent when you use the more stringent criteria of looking for ORFs that have 14 nucleotides, perfect complementarity. Okay, so while it seemed like the genes that we've identified, that this extent of complementarity to tRNA introns was unlikely to be due to chance alone, Paulo wanted to put statistical power to these findings. And so what he did first was he generated essentially scrambled tRNA ILU introns. So he made 1,000 randomly generated sequences that have the same length and GC content as the tRNA ILU intron. And remarkably, what he found was that less than 2% of these scrambled tRNA ILU intron sequences have complementarity to more ORFs than the authentic tRNA ILU intron. And just to show for comparison, this is not the case when you look for matching sequences rather than complementary sequences. Okay, then next he did the reverse, and instead of scrambling the intron sequence, Paulo scrambled the codons in yeast. So he generated 1,000 scrambled yeast genomes that have the same codon usage as the authentic yeast genome, but the codons have just been scrambled. And when he does this, he finds that 98% of these codon scrambled genomes have less complementarity than the authentic yeast genome. And so this is showing us that the tRNA ILU intron complementarity has a strong dependency on the natural codon arrangement of the yeast genome. So to summarize the bioinformatic analyses, we found that tRNA introns possess substantial complementarity to ORFs, and the extent of complementarity for both the tRNA ILU intron as well as the tRNA trip intron is exceptional relative to random sequences, matching sequences, mature tRNAs, and the UTRs. So given all of this, we wanted to then determine whether there is a sequence-dependent function of tRNA introns. And we started with the hypothesis that tRNA introns could function like microRNAs. So maybe that they bind to messenger RNAs and target those messages for degradation. So in order to test this hypothesis, we used a few different approaches. So our first approach was to generate a yeast strain that lacks the tRNA ILU introns. And then we looked at messenger RNA levels using RT-qPCR in both wild-type cells and our intron deletion strain for genes that have complementarity to the tRNA ILU intron relative to genes lacking complementarity. So this graph here is showing you the changes in mRNA levels for genes that have complementarity to the tRNA ILU intron. And so along the x-axis, you see the genes that have complementarity, and then the mRNA levels are shown for the tRNA ILU intron deletion strain, which is represented by the green dots. And these are expressed relative to mRNA levels in wild-type cells, which are shown by the black dots, and they are set at 1 for each of the genes. And so as we would expect, if the tRNA ILU intron functions like a microRNA, if we delete the tRNA ILU introns, we would expect to see an increase in mRNA levels for genes that have complementarity. And that's exactly what we see. If we compare these results to a randomly selected set of genes that lack complementarity, which are shown here on the bottom, and also shown here from this summary data here on the right, where each dot represents the change in mRNA levels for a different gene, you can see that in cells that lack complementarity to the tRNA ILU intron, genes that have complementarity show a statistically significant increase in mRNA levels relative to genes that lack complementarity. Now, I just want to point out that we do see that there is an increase in mRNA levels for genes that lack complementarity in the deletion strain relative to wild-type cells. So looking at the red bar relative to the dotted line, we think that these are likely due to altered cellular levels of RNAs in our intron deletion strain, and that could artifactually increase mRNA levels when we standardize our results to total RNA. So to account for this, in all of our experiments, we've done our analyses by comparing genes that have complementarity to genes that lack complementarity rather than comparing between strains. Okay, and then for the sake of time, I'm not going to show the data, but we confirmed that the effects that we are seeing are indeed due to the intron, because if we add a plasmid that has the tRNA ILU with the intron, we can return mRNA levels back to wild-type levels. But if we add a plasmid that lacks the intron, we can't restore these mRNA levels. So overall, from these experiments, we see that the tRNA ILU intron has an inhibitory effect on mRNA levels that's dependent on sequence complementarity, much like a microRNA. Okay, so our second approach was to use to inducibly overexpress the tRNA ILU intron. And the way that we did this was we generated a 2-plasmid TET-on system. So in the presence of doxycycline, the reverse tetracycline transactivator would bind to the tetracycline response element. And then that would lead to RNA polymerase II mediated transcription of the T7 polymerase gene. T7 polymerase then binds to the promoter of the second plasmid, and then that leads to the overexpression of the tRNA ILU intron. And I'll just note that this intron has been modified slightly at both the 5' and 3' ends to increase its expression. So in the graph here on the right, what you can see is each dot showing the changes in mRNA levels following DOX treatment for a different gene, either bearing complementarity or lacking complementarity to the tRNA ILU intron. And what we see is that in wild type cells, when we treat cells with DOX, the DOX treatment induces about a 10-fold increase in the tRNA ILU intron levels. And what we see is that mRNA levels for genes that have complementarity to the tRNA ILU intron are decreased as compared to genes lacking complementarity. We also looked in our tRNA ILU intron deletion strain, and what we see is that we get an even greater effect in the deletion strain, and this is likely due to the fact that there is no endogenous intron in these cells. So these results further support our microRNA model that tRNA ILU introns function to downregulate mRNA levels for genes that have complementarity. Now, we wanted to demonstrate that the intron is directly binding to complementary targets. So what we did is we overexpressed either a wild-type tRNA ILU intron, as in the previous slide, or we overexpressed a tRNA ILU intron that has mutations to disrupt complementarity to certain subsets of genes. And so in the first mutant, you can see that the nucleotides 25 to 33 of the tRNA ILU intron were changed. The second mutant, nucleotides 30 to 38 were changed. And so what we can see here from these graphs is that, so these are genes that have complementarity that's not been disrupted by either of these two mutant constructs. And what we see is when we treat cells with DOX, we get a decrease in mRNA levels, similarly to what we see in wild-type cells, or cells expressing the wild-type intron. If we look at cells where we have disrupted the complementarity in these intron mutants, and you can see the disruption of complementarity for each of these genes is shown in those little cartoons at the bottom with the red and the gray lines representing the disruption of complementarity. We see that the decrease in mRNA levels is now abolished. So these results show that the tRNA ILU intron is directly interacting with its complementary targets. And I won't show the data for this for the sake of time, but using this system, we were also able to show that if we disrupt mRNA decay pathways, this DOX-induced decrease in mRNA levels could be abolished. So this is showing us that together, the tRNA ILU intron is directly binding to messenger RNAs, and that is leading to their turnover. Okay, so these first two approaches, they were great in giving us a lot of information about how this tRNA ILU intron is functioning, but what they were unable to determine was whether the effects that we're looking at were due to the intron-containing tRNA or the FitRNA, so the free intron. The other thing is that we wanted to look at endogenously generated intron, and then we also wanted to see if the results that we were looking at were not just true for the tRNA ILU intron, but if they also held true for other tRNA introns. So what we did is we used 3 different yeast mutant strains, and that allowed us to use endogenously generated tRNA ILU intron, as well as look at tRNA trip intron, and then to determine whether the effects we're seeing on mRNA levels for genes bearing complementarity are due to the intron-containing tRNA or the free tRNA intron. And so I'm going to summarize the results using this chart here. So the first strain we looked at was a Tom70 deletion strain. And so this strain has decreased tRNA splicing efficiency. The second strain we used was a loss1 deletion strain, and this strain is defective in precursor tRNA nuclear export. And so in both of these strains, what you get is an increase in intron-containing tRNA levels, as well as an increase in the FitRNA levels. And then when we looked at mRNA levels in these strains for genes that have complementarity relative to genes lacking complementarity, what we see is there's a decrease in mRNA levels for genes bearing complementarity to both the tRNA ILU intron as well as the tRNA trip intron. Okay, so then we used a third strain. This is the SEN2-1 strain. So SEN2 is one of the 4 subunits of the tRNA splicing endonuclease complex that removes the intron from tRNAs. And since SEN2 is an essential gene, we used a temperature-sensitive mutant. And so when we take our SEN2-1 cells and shift them to the non-permissive temperature, we can get an inhibition of tRNA splicing. And what that causes is an increase in the intron-containing tRNAs, but not an increase in FitRNAs. And in fact, we see actually a decrease in FitRNA levels. When we look at mRNA levels for genes that have complementarity to either the tRNA trip or the tRNA ILU introns, what we see is an increase in mRNA levels for these complementarity-bearing genes. So together, these results show us that the endogenously generated free tRNA introns, these FitRNAs, and not the intron-containing tRNAs, are mediating these complementarity-dependent decreases in mRNA levels. Okay, so taking all of this together, we think that we have these FitRNAs that are functioning as negative regulators of gene expression by base pairing to target messenger RNAs, and then that's leading to their decay. And again, this is much like what we see for microRNAs. However, unlike microRNAs, we know that this process is not mediated by Argonaute proteins, and that's because Cerevisiae actually lack Argonaute genes in their genome. So this process could be aided by a currently unknown protein, or it could be occurring in a protein unassisted manner. And this is something we are currently in the process of determining. Now, given that FitRNA levels are generally very low in the cell under standard growth conditions, we wanted to see if various types of stress conditions can cause increase in FitRNA levels, and then see if that impacts mRNA levels for genes that have complementarity. And so this work was done initially by two undergraduates in the lab, Alicia and Lauren, and then has since been followed up and continued by Sarah Metcalf. So given that so what we were able to do was identify a few different stressors that impact FitRNA levels, and I'm going to show you the results for one of them, which is oxidative stress induced by hydrogen peroxide treatment. So what you're looking at here is a northern blot showing the different species of the tRNA trip. And so in the box that's highlighted, you can see the accumulation of the free tRNA trip intron in response to increasing time of treatment with hydrogen peroxide. And this is compared to the lane on the right, in which these cells were untreated. And what you're seeing is, again, this massive accumulation is about a 20-fold increase in the amount of free tRNA trip intron, despite the fact that you're not seeing any new transcript production. It's new transcript production is actually essentially turned off by as early as 30 minutes, as represented by those P and I bands at the top. And this is quantitated here for the tRNA trip intron, which is shown in pink, and then you can look at the levels of the other FitRNAs for the other tRNA families. And what's interesting is that you see tRNA family-specific differences in accumulation of FitRNAs in response to oxidative stress. And so with the exception of the LU-CAA intron, the FitRNA levels for most of the other tRNA families are unaffected or only modestly affected by hydrogen peroxide treatment. Okay, so then we want to look to see if mRNA levels for genes that have complementarity to the tRNA trip intron are reduced in response to hydrogen peroxide treatment. And so when we compared mRNA levels in cells that are treated with hydrogen peroxide to those that were not treated, we see that mRNA levels for genes that have complementarity to the tRNA trip intron are decreased relative to genes lacking complementarity, and we don't see this for genes that have complementarity to the tRNA ILU intron. So this is indicating that oxidative stress is inducing the accumulation of FitRNA trip, and that is then eliciting a decrease in mRNA levels for genes that bear complementarity. So our overall model is that we see that tRNA introns, these FitRNAs, are a novel class of small non-coding RNAs, and we're proposing that they are regulating steady-state mRNA expression by binding to messenger RNAs through sequence complementarity, and that's leading to mRNA decay. And because under basal conditions, we know that FitRNA levels are low, inhibition of gene expression is likely pretty modest. However, in response to stress conditions, we can get an accumulation of FitRNAs in the cell, And then this can cause a downregulation of mRNA levels for genes that bear complementarity. And it's possible that this is perhaps an adaptive response to cellular stress. So with that, I would like to acknowledge my mentor, Dr. Anita Hopper, as well as the current and past members of the Hopper Lab, especially those who've contributed to this work that are highlighted in blue. And then I'd also like to thank our funding in the Center of RNA Biology here at Ohio State. And I'm happy to take any questions, and you're also welcome to contact me. I've provided my email here. So thank you.
All right. Thanks, Gina. That was a great talk. I think for the sake of time, so that Dr. Tang has time for his presentation, if you would just answer questions by typing the answers into the Q&A box so that we can continue on with his presentation, that would be great. Oh, sorry, there are 2 minutes actually, so I'll pick maybe one question for you to answer. So one question, are the sequences in ORFs that are complementary to FitRNA also more conserved? Are there any codon preferences in these ORF complementary sequences, like bias to the isoleucine codon?
So we don't see any bias towards codon. So we've looked for genes that have complementarity to the trip intron and the IO intron, and we don't see any bias for that. As far as we've tried to look for compensatory mutations in the messenger RNA sequences where there's complementarity in the ORFs, and this has been a little bit difficult to interpret because we have so many genes that are potentially affected by these by this complementarity to these FitRNAs. So we're going to need to kind of establish the rules of complementarity before we can really kind of get a good understanding of that.
Okay, maybe we have time for one more. So what is the advantage of using tRNA intron to regulate mRNA?
Yeah, so one of the things that has been postulated is that small non-coding RNAs like tRNA fragments may have actually evolved prior to RNA interference. And so it's possible that this might have been an early way to evolutionary way for RNA interference, and that it has since become more specialized by using systems like microRNAs and piRNA. So that's kind of one of the thoughts of using the introns.
Okay, so now I think we'll move on to Dr. Tang's talk, but if you want to continue answering those questions throughout his talk, you can do that. So